we will now head to our next speaker. So coming up next, we have Aranzazu Arias Rojas from the Broad Institute of MIT and Harvard, who's going to talk a bit about infection-induced elevation of gut glycosaminoglycans, which foster microbiota expansion in Drosophila. Thank you so much for being here, and we're looking forward to hearing from you. Again, any questions, feel free to put these in the Q&A.
Yeah. Hey, thank you so much for the nice presentation. So just like some information before I start, like this is the work that I have done during my PhD. So this is not a perform or done at the Broad Institute. That's something I did back during my PhD, and it was kind of like my pet project, or I like to call it like that, because it was like a side project that I started with a really early observation, and then it ended up like being a really nice story. So I'm really happy to present that today, and also for the opportunity with the organizers for giving the chance to talk about it today. So thank you so much for also sharing the title of my talk. So today I'm gonna talk you about like how the host microbe interactions take place within the gut of Drosophila, and how we can learn what we can learn about that, and how we can then transfer it to different animal models. So far, like I would like to start with one of my favorite quotes for one of my favorite biologists, which is Lynn Margulis. That it comes from the book that she wrote with her son, which is Microcosmos, and it basically depict like how the host microbe interactions started when the microbes decided to colonize the guts of the moving animals or the migrating animals. And then since then we have been trying to understand why did they do that, and what is the benefit for the host, and how can we get more information about the biology through learning from the microbes and also from the host at the same time. So since then, plenty studies have been describing how microbes help or boost the health or long or make the lifespan of the host to last longer. So, for example, we have the case of Drosophila, when specific microbes like colonize the gut, enhance the lifespan of Drosophila in comparison with the germ-free paralogs. And also we have like in some other mammals like mice, how the presence of, in this case, lactobacillus enhance also the growth of the bones, and then the fixation of some of calcium. And finally, one of my main interests, which is like how the gut microbiome can enhance the immune response, and specifically why do they do that, like why they will boost the immune responses, and what are the molecular mechanisms on all of this. So all of these three different areas of research of host microbe interaction have been extremely challenging to answer. Why? Because first of all, we have like a lot of variability among the different microbes, like so a specific host, even with a certain or a given genotype or a different genome conformation or architecture, can carry different colonies, different microbe communities, right? So then that makes like the study of the host microbe interactions quite challenging. And not only that, we on top of that have the diversity in the genome conformation of the host. So that is extremely challenging, because if you really want to answer the question in how a microbe can help or boost the health of individual, then you have to go deeper and study what are the differences, like what are the differences in the population or differences in a specific genome architecture, and how you can indeed establish causality with the presence of a certain microbe and go deeper into the molecular mechanisms. So that's why it's extremely challenging to study this in hosts like humans. Now the tools are out there, like make this specifically in the humans more accessible or available, and a lot of efforts trying to understand this deeper. But let's start talking about like how host microbe or host microbe interactions happen among in different hosts. So we have humans, and as you can see here, and as I said, like it's quite challenging, because you have the host immune response that might be different among different hosts, and also you have the differences among the microbe communities. So as you can see here in host microbe, in the human host, you have like differences in the host microbe, but the most abundant species come from the Firmicutes family, right? And this seems like it's a trend that you can find across of the animal kingdom, and you can go from the human to going through the mouse and the fish, but lastly, like going to invertebrates like Drosophila, which also establish a like extremely good communication with microbes coming from the Firmicutes family. So interestingly, like it's also quite different how the immune system responds my established communication with microbes within the gut in different immunity levels. But one that we can be 100% sure is like how the host, the humans and the Drosophila can establish or the microbes can establish colonization with this host through like the resistance of the immune system interactions, right? So that's why we decided to then study or to break down this really complex question and study this in Drosophila as a host. And why? This pretty much because in Drosophila we have one tool that is called the DGRP lines, and if you're a mice person, I'm pretty sure that you have ever heard about the community cross, which is like a inbreed panel of different animals, in this case Drosophila, that can resemble the variability or the different of genome architecture that you can find out there. And what we did pretty much to try to understand the molecular mechanisms and how the host established colonization, the microbe established colonization within the host, it's because we use this tool called DGRP lines or Drosophila Genetic Reference Panel, and then we create the novel germ-free lines. So some of them were unable to thrive after the cleanse of all the microbes, but some like 92% of 92 lines were able to grow like in germ-free conditions. So after that, we try to address or go deeper in the host microbe mechanism by colonizing such lines with Lactobacillus plantarum, which is a natural microbiome member and also belongs with the Firmicutes family that can colonize Drosophila and humans as well, right? So after that, we wanted to see how this then the colonization was established during the homeostasis or without any challenge, and then what happened when we colonize such flies and then infect them with a natural pathogen that is also like a natural pathogen of Drosophila, an oral natural pathogen. After that, we quantified the loads of these 92 lines and then make a big data set. And with this data set, what we did was like to perform US analysis on all the phenotypes that we got, and then try to understand the mechanism going through how lactobacillus specifically, as a good representative of the of the microbiome from across of different kingdoms, can then establish colonization with Drosophila. And not only that, but also how it behaves when it has any competitor or how it behaves when it's alone. So something that is really interesting and what we found, and it also came from the early observation from my PhD, is like when we have lactobacillus in the fly without any challenges, seem like the lows remain stable. But once we challenge the system with a pathogen, so that means that we have lactobacillus, and on top of that we have a pathogen of Drosophila, we have a boost of the colonies or the loads of lactobacillus, which is quite interesting because you might expect it as the opposite, but that is giving you insight in how lactobacillus adapts to the inflamed environment within the gut. And so, as I said, like we performed genome ontology in the host site to try GWAS to try to identify specifically the genotypes that are giving us the boost of lactobacillus after the infection. So as you can see here, like we have like plenty candidates, and it was quite hard to understand this, like from every single variant. But then moving forward to try to understand that, we did a genome ontology analysis to try to narrow down like the mechanism behind the boost of lactobacillus during the infection. So something that we came across was actually the heparan sulfate pathway, and also the and acetylglucosamine metabolism process, as you can see here, which is interesting because it changes during the infection and also during the metastasis. So that's why we decided to um, focus on this mechanism. And one of the nice things about Drosophila is like we can also study in a deep genetical level, the whole site. And what we did then to understand this better was to um, knock down the genes that are responsible for the synthesis of heparan sulfate or N-acetylglucosamine metabolism. So the gene is called TTB, and what we did is knock down specifically in the gut this gene, and then uh, go again with the same workflow. We colonize flies, and then we either infect them or treat them only with sucrose. So as you can see here, we have again, like when we don't have, like when we have the presence of the TTB, heparan sulfate synthesis gene, we have here like the boost in the loads of lactobacillus. But when we knock down this gene, then we don't have the boost. So that means like there is a direct interplay within this gene and regulation of the lactobacillus load during inflammation. Interestingly, not only that, we also observed that after the colonization and then the infection with the natural pathogen of Drosophila, we have an overexpression of this pathway. So narrowing this down, as you can see here, like again, when we treat the when we treat the Drosophila guts with lactobacillus and then with the natural pathogen, we have overproduction of this indicated by the heparan sulfate antibody here in green. But then when we knocked out this or knock down this pathway, we don't have any responses. And on top of that, we barely have colonization of lactobacillus in the gut. So for what is lactobacillus then in the other side using this heparan sulfate, right? So interestingly, we could see that lacto cannot use heparan sulfate as it is. So it uses like derived sugars from heparan sulfate, like in this case, for example, N-acetylglucosamine, which is a sugar that is like building up the structure of heparan sulfate. But not only that, like uptake this sugar and then seems like it's using it to thrive. As you can see here, like for example, we have MRS, which is the media where lactobacillus grow, and seems like we don't have differences with the normal MRS media when we supplement it with glucose or with heparan sulfate. But when we then treat the cultures with the drip sugar, which is N-acetylglucosamine, we do have a boost in the growth. But then if it's not using it, if it is using heparan sulfate as a carbon source, like why is then using in first place heparan sulfate? What is like how long it takes for lactobacillus to uptake these sugars out of the heparan sulfate, and what is lacto use in heparan sulfate in first place? So what we could come across is that lactobacillus use heparan sulfate to build biofilms. So in our results in vitro, you can see here how lactobacillus is able to produce or form biofilms in the surface of our in vitro plates. But this is not the case, for example, for other bacteria like ECC15, which is the natural pathogen that we use for infection and is at the same time boosting the production of heparan sulfate within the gut of Drosophila. So again, as you can see here, lactobacillus is able to use this glycan to attach to the surfaces. So then going deeper in this, we wanted to see how this happened in vivo, if lacto is able to form them biofilms within the gut after the infection. So first of all, what we did was measure the heparan sulfate production, the protein production in those guts. So again, when we have when we have like the normal guts without any treatment, we don't seem like we have like a normal condition of heparan sulfate synthesis. Then when we infect those slides, seems like we have a boost in the production of this glycan. When we only colonize, seems like we don't have activation of the heparan sulfate pathway. So again, when we have two of these two of these microbes, the commensal lactobacillus and the ECC15, which is the pathogen, we have a boost in the production. And that's something that you can see here also through the gut of Drosophila, which is really nice because since the gut of Drosophila is transparent, you can perform like really good microscopy studies. So here you have green the heparan sulfate and lactobacillus in red, which is the M. cherry production, the M. cherry strain of lactobacillus. So as you can see here, like during the sucrose supplementation, we have like mainly lactobacillus going through the late part of the gut. But then when we infect, seems like the lactobacillus is spread all over the place. And this seems like it's pretty much because the presence of heparan sulfate. Because as you can see here, we have like a really low quantity of heparan sulfate in the first part of the gut of Drosophila in comparison when we infect. Whether we have more synthesis of heparan sulfate, we also have more lactobacillus all among the gut of Drosophila. So again, trying to understand better the interplay and how we already know like how the host uh, provides some cues for lactobacillus to colonize all over the gut during the infection. But then we wanted to see what are the genes that lactobacillus is using to sense the presence of heparan sulfate, and not only that, how is this like important for lactobacillus? What is the fitness trait? Um, and how like and the some like pretty much the molecular mechanisms around this. So to do so, what we did is like we did a transposon library on lactobacillus, and then we screened for the biofilm formation. So since we have like a big library of 96, I think it was like 200 yeah, I think it was like 200 plates with 96 well plates, 96 wells. So we have like plenty of mutants that could like be potentially important. So then we develop a model where we measure for the biofilm formation indicated by crystal violet, or this is like pretty much a colorimetric assay. But then also we adjust by the growth of lactobacillus during these assays. So pretty much after we see how lactobacillus wild type behaves, as you can see here in the biofilm formation corrected by the growth of the bacteria, you can see how after the line you will see interesting candidates. So we then validate like most of these candidates and in vitro and then in vivo to see whether this was important for the colonization of lactobacillus during infection. And then we came across with these five candidates. So one of the candidates was targeting the redox sensing transcriptase repressor, the extracellular transglycerols, multi-antibiotic resistant regulators, a phage entry protein, and we have like a mismatch of whether we don't really know which of these genes might be important, even though we could then validate this in vivo and in vitro. So first what we did was validating how lactobacillus was using these glycans or heparan sulfate to bind or form biofilms across of the different mutants that we identified during our transposon screening. And as you can see here, most of the mutants that we identified were not able to form biofilms and neither to bind to the surface that was coated with heparan sulfate. When then we look in vivo, you can see here how, like for example, the redox protein seems like was not important for this, for the boost of lactobacillus loss after infection. But seems like all other genes seem like might be important because even if we don't have significant differences, like the loads just go down and we're unable to thrive during the infection conditions, which then resembles like the overall phenotype. So lactobacillus therefore is not able to thrive because it lacks, for example, for like the T gases like transglycosylate, and this is pretty much because then it's unable to sense the presence of heparan sulfate among all the gut during the infection. And yeah, so pretty much the overall the overall take-home message of this study is that microbiota can persist. In this case, lactobacillus can persist in the inflamed environment. And this is because it can acquire not only resistance, but also can acquire new nutrients in order to expand and grow and thrive in the inflamed environment because they are potentially changing all the time. And in the other hand, in the hand of the host, we know the heparan sulfate pathway facilitates the colonization of lactobacillus by enabling the adaptation to uh, glycosaminoglycans during the inflammation. And interestingly, also, if you would like to see this from evolutionarily point of view, unlike two pathogens, microbes like lactobacillus can thrive. So what is one of the things that we also observe is like this is like really coupled with the host because the more loads of lactobacillus that you get within the gut of Drosophila, and the lifespan of the fly is also longer. So that means that this is really tied to the host fitness as well. Whether lactobacillus will colonize all the niches that are available within the gut during the inflammation to avoid the colonization of a secondary colonizer, in this case pathogens. So many other questions are remaining in this study, but this is like what we have just published, I think like last year. Yeah. So then if you're more interested about this, like you can just find this thing online with the same title. And I'm really happy to take your questions. So thank you so much.
Wow, that was an amazing wrap up of your side project. Congrats on the publication and well done. That was amazing. We definitely do have some questions in the chat, but I just want to start off with something that kind of mirrors what Eugenio was asking. Um, I'm super impressed by the images you took of the Drosophila gut along the way. Just as someone who works in mice, the scaling of Drosophila is kind of hard to imagine. So I was kind of wondering what the challenges of using that specific model were, and that kind of ties into what Eugenio is asking is if you maybe are planning to do some testing in mammalian models or other types of models for these tests.
Yeah. I mean, currently in different kind of questions right now during my postdoc. So I don't know. I have heard from my previous lab that they are trying to pursue a different story or a side story on this, but they work primarily with flies, right? So then I think like who they will stick with flies. So, but it will be really really interesting if somebody like try to pursue the same kind of question and like mammals. So I know
Mm-hmm.
that there are like significant efforts to try to understand this mechanism also in fish,
Okay.
zebrafish, by Karen Guillemin at the McGill University. So then I think like there like seems like this is really conserved among like different hosts. It would be amazing if somebody tries to like to study this in mammals. Like yeah, it would be great.
Passing it to the next person for sure. Amazing. We also have a question from Eleanor in the chat. Eleanor is asking that infection with unrelated pathogens boosts the bacillus with the heparan sulfate increasing the available biofilm. What does this mean for the gut immune system? If you can comment on that a bit.
Yeah, that's a really good question. So then one of the results that we also got, like we have in flies we have NF-kappa beta signaling as well, right? So we could identify like the NF-kappa beta is regulating the heparan sulfate pathway because when we knock out the NF-kappa beta in flies, we don't longer have this effect. So which means like
Mm,
it's extremely tied to the host immune system. And not only that, like seems like it's also regulating the intestinal stem cell production, and which then also helps to like to renew the whole gut barrier in within the gut of Drosophila. So it seems like whether like when the host is sensing the the pathogen through the NF-kappa beta or any of the receptors like TLRs and in the gut. So then it's promoting the synthesis of heparan sulfate. So that means that yeah, I don't know if I'm answering the question of if it's taken just if I'm just yeah. The cells growth, so then does the unrelated pathogen after the local immune response to permit pathology? Yeah, so then yeah, like it's extremely tight, like when it comes about infection. So then after sensing the infection, there is more production of heparan sulfate. But then something that I would like to clarify is like when you knock out also the NF-kappa beta or the primary immune response in Drosophila, or it's called the intestinal deficient pathway, IMD pathway, you also boost the loads of lactobacillus. So I'm not pretty sure what is going on. I'm pretty sure like it's because you don't have any immune pathway that is regulating the control of bacteria. So then therefore the host like guts like nuts, and then it's just like a lot of bacteria within the host. But yeah. So I'm sorry. I don't know if I answered the
No,
question.
that was great. Thank you so much for answering that. Well done. So thank you so much. I think there may be a couple other questions in the chat, but a lot of people just applauding how amazing your talk was. So congratulations again, and thank you so much for sharing that work with us. Um, Make sure to check out that publication online. And yeah, if you're able to stick around and just see if there are any other questions in the chat, that'd be
Yeah,
awesome. Thank you
great.
so much
So
and enjoy the
much.
rest of your day.
Yes, thank you.